Right now we will ask Professor Paul Thompson, who will represent my university, University of South California, and his topic is the Enigma project investigating brain disease with imaging and genetics in 50,000 people. Пол Томпсон, директор мирового консорциума, объединяющий порядка 185 институтов, 30, теперь уже 7 стран. Россия включена, Украина, Беларусь и Казахстан. И он сегодня представит нам материалы своих исследований. Please, Paul.
Здравствуйте и доброе утро. Благодарю вас, профессор Зелман. Извините, я говорил по-английски сейчас. So, thank you very much for the invitation this morning to speak. I'll tell you a little bit about a project that involves a lot of scientists around the world in mapping the human brain. And I'm very grateful to my colleague, Professor Zellman, who introduced me in engaging a very large number of Russian scientists from across the Russian Federation in this joint activity. The topic is called Enigma, и по-русски это загадка, международное сотрудничество по исследованию генетического предрасположенности к болезням головного мозга. And it's a joint effort led from the University of Southern California, but involves a lot of institutions around the world. So, really just to motivate this topic a little bit, if you think about how you could map the human brain in different ways, we really have a few different methods available to us for looking at brain structure and function. So, to look at the anatomy, you can do an MRI scan, a three-dimensional MRI scan with computed tomography. To look at the molecular content of the brain, you can look at PET or SPECT, or with more modern approaches, looking at blood flow using functional MRI. And then also, as in Dr. Lebedev's talk, you can look at very fine-scale electrophysiological activity, either directly by imaging the cortex with optical intrinsic signal imaging or with EEG. And a lot of the interest is trying to relate these measures to other branches of science. And so you can imagine also looking at the genetic code, trying to understand how the genetic code might affect these signals. But this type of question has traditionally not been answerable using techniques that we have so far. So I'll talk about a little bit how to get into it. So you'll hear nationally there are initiatives to map the human brain, biobanks that contain data from brain imaging and genomics. And in 2009, Enigma, which is a consortium, started the largest brain imaging study in the world, largest in terms of the number of scientists involved, the number of people who'd been scanned, and also the extent of the data that was available to look at these questions. The main goal of Enigma is to try and answer the question, what factors help or harm the human brain? Collectively around the world, we're analyzing brain scans and genomic data, DNA, from over 53,000 people. And involved in this project are people from 35 different countries, over 500 scientists in 30 different working groups looking at different questions. And this has led to some really quite high-impact papers in neuroscience, in Nature Genetics and Nature this year, mainly looking initially at 12 different brain diseases. And so if you think of the different diseases that affect the human brain, within psychiatry, there's schizophrenia, depression, bipolar disorder, post-traumatic stress disorder, obsessive-compulsive disorder. Also learning disorders in children, notably autism, attention deficit disorder, and also brain disorders that you wouldn't traditionally think of as being within neurology, addiction to drugs and alcohol, infectious diseases like HIV that also affect the brain, and also within the dementias, Parkinson's disease and Alzheimer's. So this is where the data comes from. Within the Russian Federation, we have sites at the Child Health Center in Moscow, the Institute of Neurology in Moscow, also at Akadem Godorok in Novosibirsk. There are a number of centers contributing data. And what this picture shows is the different diseases that these centers study. So each of these centers collects brain images, and each of these centers collects DNA from people. And the different diseases are listed here: autism, schizophrenia, epilepsy, many different disorders of the brain. And the question becomes, how do you see patterns? How do you find trends and patterns in this data, even though it was collected with different goals in mind? So this has led to really quite remarkable partnerships across the world, often papers with over 300 authors, as in particle physics, if you remember the paper on the Higgs boson. There's very large numbers of people involved. And the work's organized into different topics. So there's questions that look at the brain from the standpoint of genetics, projects that look at the brain's connectivity, how the brain is connected, and then also particular working groups looking at different disorders. We mentioned in psychiatry these different disorders. And there's a lot of interest in mathematics to try and compare patterns in the brain from one disorder to another. Now, this provides an interesting opportunity. If you have this large number of brain images, you can begin to look at linkages between the genetic code and the images, because there's so much data. Even though the effects are very, very tiny, you can pick them up. So this leads to the notion of cracking the brain's genetic code. Can we use all this data to understand how the DNA affects the brain's signals? Now, one reason it's called Enigma is after a project in the Second World War related to the Enigma machine. And so the Enigma machine was an encryption device used by the Germans to encrypt radio messages. And the Allied forces during the war developed a decoding machine to try and decrypt these signals and turn them back into language. Now, the reason it's called Enigma is that the original method to do this was by brute force. It was by permutation. You tried every possible code. But the insight is that if you could understand the principle or the model or the axioms of the code, you could do this much more efficiently. And I think one of the challenges in Enigma is trying to understand collectively how these signals are encoded in the human brain. So, to allow people to share data internationally, a neurosurgeon called Jean Talarac in France in the 1960s developed a three-dimensional coordinate system for brain data. So suppose you and I had scanned a person with MRI on the right there, but we wanted to refer to the information in the brain using a 3D coordinate system. He developed a method to stereotaxically map it into a coordinate system, and you could map post-mortem data, functional data, molecular data into these 3D coordinates and make associations. And you could begin to compile a population of information in these 3D coordinates. So let me give you an example. If you take a brain scan and you use a computer algorithm to separate the brain scan into gray matter, the neuronal part of the brain, white matter and fluid, you can say, suppose a patient has Alzheimer's disease, Alzheimer po-ruski. Is there a pattern in how people with Alzheimer's disease differ in the brain? And you can measure the thickness of the gray matter, how much cortical gray matter there is. And if you collect data from a large number of patients and from a large number of healthy people, you can say, well, in Alzheimer's disease, these regions of the brain, the memory systems, the association systems, are reduced in the level of gray matter by about 10%, on average about 10%, but not the whole brain, just some parts of the brain. Now, if you were to scan people with Alzheimer's disease when they arrive at the clinic, 6 months later, 6 months later, 6 months later, you'd see a progressive decline in the brain. Their memory function would decline, their language would decline, their ability to perform socially would get worse. And if you compare these brain scans and the thickness of the gray matter of the brain with healthy people, you see this spreading wave of loss of brain tissue. And you can reconstruct this. And so from a database of brain scans, you can say, at a certain stage of the disease, we expect to see this amount of tissue and compare them with healthy people. Now, this is used commercially in clinical trials. So if you develop a new drug or a new medicine to protect the brain from tissue loss, you can collect brain scans, MRI, MRT, from people who have been taking the new drug. In this case, olanzapine is a new drug to save brain tissue. And you can compare the rate of loss of tissue between the new drug and the existing drug. So an existing drug, haloperidol, is used in schizophrenia. And you can begin to say the new drug slows the rate of tissue loss, at least in a population the average rate of tissue loss is less, and you can begin to do head-to-head comparisons of these medicines. Now, medicine is not the only thing that affects the content of the brain. You can begin to think of other factors, and so stress, high levels of stress, are related to the level of cortisol, a hormone in the blood. And if you measure it and make a statistical map of the association between the level of stress hormone in the blood and the thickness of the cortical gray matter, you will find patterns of linkage. The level of stress is associated with reduction in the gray matter. And you could also begin to look at the genetic code. So you could say there is a gene that 1% of us carry a very bad genetic variant. This gene is called TREM2. It's a gene that increases the risk of Alzheimer's disease. And you could say, collect brain scans from all the people who have the bad form of this gene, and you'll notice that they lose brain tissue more quickly. So you could begin to imagine mining the human genome or mining any other source of information for predictors of how fast the brain is aging, or whether there are any genes that protect us, or whether there are any medications that protect us. So let's formulate this as a mathematical problem. You could look at the DNA, and you could look at brain scans, or you could look at the symptoms of a disease. And at each location in the human genome, you could ask this question: is there any predictive ability of this point in the human genome in relation to the function or the structure of the human brain? And this is already done. So in psychiatry, people collect DNA from people with depression, schizophrenia, Alzheimer's disease, and they have found markers that increase the likelihood that you will get these diseases. And in particular, there are areas of the human genome called polymorphisms. So all of us in this room have fundamentally the same genetic code, but there are locations in the DNA where each of us has a different letter, a different nucleotide. And very cheaply, we can collect this information. So a sample of saliva, you could spit into a test tube, and within a couple of weeks, you can find 1 million nucleotides of your DNA, and you can even get your personal genetic code. There are many companies in the Russian Federation, in the United States, Illumina Genetics, or 23andMe is a company that will give you 1 million nucleotides of your genetic code. So you can begin to imagine collecting a very large amount of this data. So suppose every one of us has a genetic code that we know, and it isn't the whole thing, it's just these points where there are common variants in the genetic code. And suppose there was a genetic code that gave us a large brain. Now, maybe controversial, but if that were true, you could march along like the Enigma machine and find a point in the genome that statistically predicted the size or the function or the connectivity of the brain at that location. And the way that geneticists show this information is on the right here. So you look at different genotypes. So each of us has 2 chromosomes. You might have a letter C and a letter C, or you might have a letter A and a letter C, or a letter A and a letter A. There are 3 possibilities. And so each of us in this room, at each of those markers on the genome, will have one of those 3 possibilities. If carrying one of those genotypes is statistically associated with a larger brain or a different brain function, you can plot it in the plot on the right. Those sectors are chromosomes. There are 23 chromosomes. And if there is a genetic location that predicts the brain signal, the p-value, or the significance of the association, is plotted with higher points being more evidence. And so this is known as a Manhattan plot in genetics. So this is what we do in Enigma. All around the world, people are collecting MRI scans of the human brain. 185 institutions. Using computer programs, we measure, first of all, the size of different parts of the brain. Using a different type of scan, diffusion tensor imaging, we look at the integrity, the fiber architecture of different tracks, different connections in the brain. And then in genetic analyses, we test statistical associations between genetic markers and these signals. And you can imagine if you have 1 million genetic markers, you could do this 1 million times. And because people are in different parts of the world, we perform a meta-analysis. And we aggregate the evidence from each cohort, each population, and the weight that each population gets depends on the size of the sample. So if you've collected 4,000 brain scans, that gets a lot of weight. If you're an imaging group who has collected 10 scans, you contribute to the analysis. So this is what we found. We found that if you measure the size of part of the brain that controls learning and memory, the hippocampus, po-ruski hippocamp, you'll find that there are points in the genome that have a huge impact on the size of the memory system of the brain. In fact, on chromosome 12, there is a gene that makes your brain look 3 years older. So remember, 1/3 of us in this room carry this genotype. It makes the brain look 3 years older. 2/3 of us in this room don't carry this genotype, and our brain looks younger. And similar statements can be made at different points. A Russian geneticist here in Moscow, Professor Kazima Bulayeva, discovered the function of this memory gene. She went to Dagestan, and she's a medical doctor who collects DNA from people in the ethnic isolates of the Caucasus. And she found that the same gene that Enigma had discovered was associated with psychosis, with mental retardation, and with a number of different conditions in her patients. And it shows the power of communication across the internet that she was able to make this connection with a large international study and discover the function of the brain. Now you could say, tell me which point in our DNA has the most influence on the size of the brain. And there was a study in Nature earlier this year, 300 scientists, where the gene that we each carry has a variant that affects the size of the brain by 9 cubic centimeters on average, so two teaspoons of brain tissue. The gene is called Microtubule-Associated Protein. In fact, neuroscientists have studied this gene for many years, but each of us has a different form of this gene. And it's famous. The same gene is associated with Parkinson's disease, a very terrible condition of degeneration later in life where movement is impeded in Parkinson's. So there's this notion of antagonistic pleiotropy, a gene that has survived in human populations that makes the brain bigger. Late in life also promotes Parkinson's disease. So we have this evolutionary saving of a gene that is helpful when you are young, but negative when you are old. So as this project grew, we began to have the power to see other effects on the brain than just simple genetics. So suppose you take the brain scan and you measure the size of each of the substructures of the brain. So you will see in the middle here structures involved in motor learning, structures that are involved in anger and emotion, the amygdala. There are structures involved in addiction, the nucleus accumbens. And you can say, first of all, are there any genes that affect the size of these structures? Now, the size is not the only thing. The function is probably more important. But we began to make a map of the genetic influences on the size of these structures. So for each part of the brain, you go along the genome and ask where it is in the genome that predicts the size of these structures. It turns out that 30% of the variability in the size of the structures is predictable from the saliva sample. It's incredible. It's completely incredible that the DNA genetic code that we each inherit at birth accounts for 30% of the variance in the size of these structures. Maybe quite controversial. How is the computation done? given the large amount of brain scans and the size of the data set, the computation is distributed across supercomputing centers around the world. There are training videos to help people analyze data locally and send information back to several centralized sites that pull the information. These are the cohorts. You'll see the names of the different populations studied. ADNI is American, Imogen is Western Europe. There are populations also here from Novosibirsk and the Child Health Center in Moscow. And you can see that in this type of plot, the statistical evidence that a certain gene affects the size of part of the brain is shown for different countries across the world. And for some genes, there is overwhelming evidence that that gene influences the brain. And you can do more biological studies to understand how the gene is expressed, to understand whether the particular location mapped is the only one that matters, and to look at more complex characteristics of morphology, such as the shape of the structures or other metrics. You can then go to mice. And so in the wet lab, there are homologs of human genes. The genome is very similar between mice and humans. And you can identify what feature of the cellular physiology is altered with variants in this gene. And it's remarkable that the genes that affect the size of the motor systems of the brain in humans also affect the number of cells, the size of the cells, the dendritic complexity of cells in mice. And so this gives you a way forward to understanding the mechanism in a manipulated context of outbred mice with different genomic variation. Now, given that this was possible, there were articles in Science and in The Lancet saying crowdsourcing meets neuroscience. In other words, if you want to study the brain's function, you don't have to collect all of the data yourself. You could look at all of the aggregated information at the other sites and discover factors that maybe age or protect the brain by an amount probably equivalent to about 3 years of aging, which is quite significant. So the mathematicians and physicists began to look at this problem and said, why do you just measure the size of things in the brain? You have an image. An image has 1 million observations. So you can imagine doing a test of statistical predictors at each pixel in the image. So if you imagine that the brain is made up of individual pixels, the numerical value is either the function or the molecular content of the brain, you could begin to think of a brain-wide, genome-wide test that does 1 trillion statistical tests at each point in the genome, at each point in the image, trying to discover patterns of linkage between parts of the brain and parts of the genome. There's a problem. If you do 1 trillion statistical tests, your computer memory will be full. It creates 200 terabytes of data, equivalent to 1 million brain MRI scans, which is difficult to store. Secondly, each site in Enigma may not want to send all of their medical images to the other sites. It's better to do parallel computation. It's too much of a computation to do at one site. And secondly, if a new site joins, you don't want to have to recompute everything that you've done so far. So when we look at the effect of a genomic marker on the brain, we look for statistical associations with the signal in each pixel. And this is a study in North America, the USA and Canada, in 700 people, all elderly. And these are the locations where one marker in the genome predicts the function of the brain. And you will see that there are very specific patterns of where the gene has an effect. This project collected more data, and you'll see similar results. A project in Norway scanned 800 people, and you'll see the same gene affects the same part of the brain. A project in Australia collected DNA and scans, and you'll see again the same regions of the brain. All of these people are different. But a large-scale worldwide computation could combine the evidence from all of the countries that are participating, and it looks like this. So when you combine data from 3,000 people, you see that this gene affects one structure of the brain. and all of the noise and statistical variation that comes from the limited sample is washed away. Now, that was one gene, and we saw that this gene affects the putamen. It's a part of the brain involved in craving, addiction, motor function, and learned behavior. Suppose that you were to go at each point along the genome and look for associations between the nucleotides and the individual points in the image. You could imagine a genetic map where you can explain around 30% of the variability in a population by testing the saliva of the individual. We have begun to set up a computation so that this is efficient. In the field of machine learning, there is a type of algorithm called multi-party private distributed computation. Suppose each of us has private information. We don't want to send the private information to everybody else. There are methods that can compute on private information, but aggregate only public information at the end of the computation. And so when we add a data set to Enigma, none of the people in Enigma shares DNA with any of the others. They just perform the computation on the data sets and discover the genes that are effective based on all the data. One method tried is called private Lasso. The mathematics says, tell me which predictors in the genome are useful in predicting brain signals. Lasso is a penalized regression method. It favors models that have a small number of parameters. And note that in our study, each institution maintains its own data. It's private. They don't share it. It stays at the site. And we're very lucky to have the partnership and help of Professor Alexander Kulyashov here in Moscow, who has developed many methods for machine learning and big data analytics. Благодарю вас. And you will see that these things are becoming problems in information theory. They're not so much problems in collecting data, they're problems in making statistical predictions from the amount of data that's available. Now, each of the people that collects the data reports to a working group leader in different countries. You'll see across Western Europe, Canada, Ireland, Africa. Some groups are in the UK. And the manuscripts look like this. You will basically say, tell me how depression affects the brain across the world, or tell me how schizophrenia affects the brain across the world. Now, you could imagine a worldwide survey that says, across the world, can we see a pattern in the brain that is diagnostic of schizophrenia, or bipolar disorder, or depression? Because in the clinic, it's often quite difficult to distinguish the symptoms of these different disorders. So essentially, you can measure those parts of the brain automatically from a scan. And Enigma found that in depression, the only part of the brain that is consistently different in a brain scan is the hippocampus. The brain differences are greater if you're depressed when you're younger. The brain differences are less if you take a medicine called Prozac, which drug companies are very interested in screening different medications. And to make this discovery, we were helped by an academician of the Russian Academy of Sciences, the Siberian branch, in Novosibirsk, Lyubomir Aftanas, who for many years has been collecting data not just on the genome, but also on the methylation, the way the genome is modified throughout life. And he has suggested that we not just look at the genome, but also the changes that occur in the genome throughout life, adding another dimension of complexity. You see him here with his colleague in high field mapping of the brain, Renard Sagdiev, who leads the MRT Institute in Akadem Godorok. And they are very helpful partners in this, looking for different methods to screen the genetic code. Also, we're fortunate to have the partnership of other institutes in Siberia, the Mishalkin Institute, where our colleague, Professor Zellman, was trained. In fact, this is Professor Zellman as a young man. These are centers which have developed a very large database of information from patients undergoing operations pre-surgically, and they also collect DNA and clinical information about recovery. So you can imagine not just linking genes with brain, but also brain patterns with the potential to recover following a surgery or recover with different types of medication. Now, drug companies are interested in developing metrics for each disease and for each medication. So you could begin to say, in 21,000 people with these different illnesses, can we develop norms? Now, of course, it's not normal to have these diseases, but there are certain confidence intervals for the structural volume and integrity in different illnesses. We have several partners across the Russian Federation. The Child Health Center here in Moscow is collecting large amounts of data from children. And last night we had a very helpful meeting where we're discussing ways of incorporating the data from the children who are scanned with MRI to compare it with the adults and see which are the structures that differ the most. In fact, here is Professor Baranov and Professor Namazova, who direct the institute, and good Russian food. It's important to recognize that MRI is very expensive. So this is a scanner at the Child Health Center in Moscow. Each scan costs in United States dollars $500. So the cost of doing a project that scans 30,000 people, it would be ridiculous. It would cost 15 million US dollars. And of course, nobody would be willing to do it. It'd be difficult to organize. And so I think the notion that rather than scanning each individual, you can look at the whole world's aggregated data is powerful. Now, other applications. This is my daughter. So when my daughter was born, she was born prematurely. She was born at a gestational age of 30 weeks. And the pediatricians are very nervous. They say, well, will this child's brain development be fine? Or are there things we can do and empirically test that get the development of the brain back within the statistical norms for a child of a different weight or a different age? So in Enigma, one project develops a lifespan map of the brain's aging. So you can imagine, it's interesting, this is the size of the brain. It's a simple metric. I am 44 years old, so I'm here. And if you are in your 30s, maybe here. These are percentiles of the world population for the size of different parts of the brain. Now, it's not particularly interesting to say I'm 10th percentile or 90th percentile, but it's very interesting in the context of neurorestoration to say this person's brain recovered from being a low percentile to a higher one. So in the end, you could test a drug treatment relative to the world aggregated data, maybe an antibiotic, maybe an antipsychotic, or maybe an anti-dementia drug, to see if the rate of brain aging was reduced. Now, from a neuroscience standpoint, we're beginning to see that the parts of the brain don't age in the same way. These curves are very different depending on the part of the brain. And you could develop metrics depending on your interest. Now, I wanted to say, I have a little bit more time, a couple of things that we have learned from a political standpoint in running this type of study. Enigma now analyzes brain data from about 50,000 people. Now, if we were to apply to the government or to a private donor to collect brain scans, it's very difficult to collect brain scans from more than 100 people. So we can see that certainly in genetics, to achieve a sample that is powerful enough to detect single variants in the genome and their effect on the brain, this type of partnership is absolutely necessary. No single government needs to buy all the scans. So we mentioned how no review panel would approve a study that collected this much data. Also, as new members join, they add computing power. They add their computer as well as the data. So there's a sort of crowdsourcing aspect. The other thing that we learned is that most centers that collect medical data don't want to send or share the data with anyone else. So there's a legal and ethical liability with sending biodata around. So the way that the algorithms are used is that they're sent to the sites where the data is. Computations are performed, and measures of statistical evidence are sent back to several coordinating sites, not just one site, but several sites. We found out that there is a distrust of coordinating centers. And so often it's better if the evidence is sent to everybody. And this is also related to legal constraints on the export of biodata. Also, the computations are private. There's some worry that DNA, if there were markers that predicted a grave risk of illness, might be used in a prejudicial way to deny medical care. And so the scientists maintain the control of their data, and they can remove it. They can say, I don't want to be in the study anymore. All of the Enigma projects are led by quite young scientists. And so the junior analysts who are expert in machine learning, mathematics, and physics are typically in their 20s and 30s developing computer programs that are then sent around the world and run. How is this funded? It was extremely difficult to convince the United States government to fund Enigma. And we said, it's critical for the trust of this project that each of the countries participating receives some funding, even though the United States taxpayers are paying for this. Now, it's difficult. I am a British person. My colleague, Dr. Zellman, is a Russian person. We were going to the American government and asking, could you send the money out of the country? The convincing argument made was from the field of mathematics. If you wanted to solve a difficult mathematical problem, no person would say, can we solve this within America? Or can we solve the mathematical problem within Australia? It doesn't make sense. The ideas for solving the problem inevitably come unexpectedly in some cases from around the world. And I think this argument from mathematics is compelling in the case where you have too much data for any one person to scrutinize. So now, for every dollar that is invested in America, it's matched by $25 outside the country. Each national government also contributes money to the scanning and analysis of the data. We have been discussing this week at Moscow Science Week international exchange. In fact, we're lucky to be able to have Mikhail and other people from Professor Kulyashov's group come back to the Russian Federation, be partners with the medical labs in Enigma, and make discoveries together. Finally, I want to talk about something a bit more futuristic. So we presented a method to screen brain scans, but we didn't look at anything interesting in the scan. We looked at the size of parts of the brain. Neuroscientists today are more interested in the principles of communication in the brain. This is an image at 7 Tesla, high magnetic field, MRI, of the neuronal pathways that connect different parts of the brain with each other. And you can see the detail that can now be achieved. So in one brain scan, there's the potential to map the entire network of communication, at least at a structural level, between regions of the brain. How is it done? Using a standard MRI scanner, rather than measure the molecular content of each pixel, you measure the direction of diffusion of water. In 1960, it was discovered by MRI physicists that when water was diffusing out of a pixel, or a sample, the MRI signal was attenuated. And the amount to which the MRI signal was attenuated had an exponential decay. And the coefficient of exponential decay depended on the speed of diffusion of water. So you can map it. So with an MRI scanner, and this is a standard scanner in a hospital, you can look at a slice of brain and watch the water flow through the neural pathways that connect one part of the brain to the other. And if you take an image a few milliseconds after an initial magnetic polarization, RF pulse, you can then compare it with the image taken a few milliseconds later and a few milliseconds later. And you can begin to see that as water spreads through the brain, there's a reliable index to the connections that communicate information from one part of the brain to the other. So this is achievable on scanners available today by following the paths of water through the brain's networks. And you can begin to think of a connectome, essentially a map of the physical connections from one part of the brain to the other, The color here indicates the direction. So green is this way, blue is this way, red is this way. And you could say, how could we make sense of it? If we collect this data from very large numbers of people, can we see patterns? Can we see things that predict how the brain is connected? Can we see genomic markers that affect the wiring and communication of the brain? Now, if you collect data from people with dementia, there's a form of dementia in late life called frontotemporal dementia. There's degeneration of the frontal lobe and the temporal lobe, and the fibers that connect them are lost. So you could say, tell me in a patient whether the number or integrity of the connections is within the statistically normal range for people of this age. Now, frontotemporal dementia at the top is different from Alzheimer's disease. In Alzheimer's disease, there's a very different pattern of loss. And you could say, let's collect a database for every pathway in the brain. Here, the language pathways. In fact, here it is. There's a pathway that connects the frontal lobe of the brain through to the speech area and the comprehension area of the brain. And at each point, you can assess the microstructure, molecular content, and synchrony of activity between the sites. So the mathematicians who have been processing this data have said, let's simplify it. Let's make it less complex and tractable from an analytic point of view. It's possible to take two regions of a brain scan and state whether there is a connection between them according to diffusion imaging. Now, there may be a connection, but the connection may be less intact or may have a depleted molecular content. So imagine if you were to make a wiring diagram or a connectivity map for your brain. So suppose you have a scan. On the top left, your MRI scan has been automatically divided into 70 regions. And you could say, in my brain, is there evidence for a connection between each piece of the brain and other pieces of the brain? And in the same way as you might have a map of cities in a country, you could express whether or not there's a connection by a 2x2 matrix. You could say, in this person, these are the areas that have connections. Or you could look at the property of a connection. Now, this is just an image. So turning this network into a two-dimensional array means that it's analyzable using similar methods to the ones that we use to analyze MRI scans. We have compacted the network into a two-dimensional image. So in Enigma, all these connectome maps every day are being mined for predictors of wiring. Now, it's possible to be a little bit more intelligent in the survey of the genome. It's possible that these connections that route information from one part of the brain to another have different degrees of genetic influence, environmental influence, different degrees of change throughout life. And it's possible to compare identical twins. Identical twins have the same DNA, and if they're more similar than randomly selected people of the same age, that connection is likely to be one that has a genetic code that affects it. So we pre-screen the connectome to look only at connections that are more similar in identical twins, and then perform the same genome-wide screen as we mentioned earlier. So you can imagine an algorithm that goes through the networks of the human brain in this matrix and goes through the genome and asks, tell me the genes that affect whether a connection is present, and tell me based on all of the scans that are collected in the 35 countries of the world that are in Enigma. When this is done, the geneticist then can find the same connection in mice. So this is quite hard to believe. A mouse has a very small brain, but it's possible to map the diffusion of water in different parts of the brain. And remember earlier we said that you could track the diffusion at different points and follow the axonal pathways in the mouse. And in the same way as the computer algorithms can compute the network of connectivity in human brains, it's possible also in the mouse. Now, the elements of the mouse brain have different proportional size, but by and large, the network's present, except for language, mice don't speak, but in terms of motor control or sensory control or vision, are largely similar. This is a connectome of a mouse. The dimension of the mouse brain is no more than ½ an inch across, and in ½ an inch, we have expressed graphically the entire network of the mouse. This scan takes 24 hours. So no human would sit still in a brain scanner for 24 hours. A mouse, you can get much better quality data. Now, we mentioned that Enigma is a little bit like decoding the Enigma machine. The reason the Enigma code was cracked was not that there was a powerful machine. It was that a team of mathematicians determined the principles or the axioms that governed the generation of the code. So there are many efforts today, this is one example, to take the connection map of the human brain, think of it as a network like the internet, and compute measures of network complexity, network modularity, the topology of the network, and feed them into machine learning methods such as support vector machines, more modern methods, deep learning, and say which properties of the network are different in different diseases. It may be that the way in which we divide the brain is incorrect. There may be a partition of the brain that correctly represents the elements of the brain that are interconnected. So it's possible to run a computer algorithm that randomly segments the brain into hundreds of regions, examines the connections between the brain regions, and determines whether that network map is predictive of a certain disease. Now, this is data mining. You essentially have no model of connectivity, and you try them and see how predictive they are and rank them. There are branches of mathematics that have to do with the understanding of types of networks and their topology. So rather than have to search all of the ways you can subdivide the brain, and you very rapidly get intractable mathematical representations of the brain network, you can divide them, and the computer algorithms can search them in an efficient way that prioritizes the information. And these are useful. So if you want to predict if a person is depressed, has depression, or predict if a person has Alzheimer's disease, based on the pattern of wiring alone, you have about 92% accuracy in diagnosing Alzheimer's disease. Alzheimer's disease is not difficult to diagnose. Depression, you have about 80% accuracy in diagnosing if a person is depressed based on the anatomical connections. Now that's interesting. You would think that depression is a functional state. So the fact that the network can predict it is interesting. So to summarize, one lesson I think we have learned through Enigma is that it's possible to screen information worldwide and find very small effects. For example, none of the predictors in the human genome individually affect the brain by more than 1%. So if you think of all of the human natural variation in the brain signals, we didn't find any markers in the genome that affected those markers by more than 1% of the variance. In aggregate across the genome explains about 30% of the variation. And this is why the search was difficult. Obviously, this predictive model requires information from across the DNA. I think the notion that you could solve big data questions using crowdsourcing is an interesting one. So doing a search of the DNA and images and networks obviously requires large-scale computation, but this can be accelerated by worldwide distributed computation. And so this is a principle of computation that has been very effective in analyzing this type of data. Now, the data is not the only thing. This would not have been possible at all without the kindness and willingness of investigators in different parts of the world to compute on data that took them many years to collect. And so you can see here, there are groups, for example, in Bangkok. This group does MRI scanning of children who are infected with the HIV virus. So in Thailand, it's a very prevalent infectious disease to have HIV. When they collected the data, they had no intention of sharing it with people in Novosibirsk, or in Moscow, or in Italy, or in Amsterdam. But I think there's a genuine feeling of excitement that this type of information collected for different purposes could be useful to all of us. An acknowledgement is quite impossible because of the number of centers involved, but particularly we're grateful to those institutions that have contributed mathematics and medical data. These are my students in Los Angeles. And if you want to join, it's not necessary that you be a medical person. If you are a mathematician or a neuroscientist or a geneticist, and you have an interesting idea for something that could be tested, it's quite possible to post this idea on the website about Enigma, and we will try and generate a team of people who can help you to test this. And if you go to this website here and just press members, all of you are welcome to join in. We need your ideas, and we may be able to take this forward in a direction that is today unimagined. So thank you very much. Spasibo большое.
And what is the size of a control group? Yes?
That's a very good question. So the question is, what is the size of the control group, the healthy people, compared to the patients? We basically used whatever data people had, and it turns out that on the whole, 1/3 of the people in Enigma are patients. 2/3 of the people are healthy. Now you could say, why are so many healthy people being scanned? The reason is, many studies of the genetics of brain only look at healthy people. So, for example, the Human Brain Project is trying to develop a normative database of healthy people. The UK Biobank in England is healthy people. So we didn't design this, it was just whatever data people had. Now, having said that, the patients are about 1/3, and they're represented in approximately the incidence of those diseases. And so depression in the United States, for example, is thought to affect 30% of people. It seems high. So if you wanted to study depression, 1/3 times 30%, there would be, let's say, 5,000 people that had that type of information. But the rarer diseases are obviously a very small fraction of the overall data. You had a second question.
Thank you. The second question is the following. You have enormous data of patients, yes? And do you see any statistically significant differences between brains of different populations? I mean, Chinese, Russians, Americans, and so on.
So the question is whether there are differences between people in different countries or populations or of different ethnicity. This question has been quite politically inflammatory for probably 1,000 years and is very interesting. So in the United States, it's politically very difficult to ask this. And if you try and address it in a grant, the reviewers will go crazy. But it's medically important because we know that the risk genes for certain brain diseases and many diseases are not uniformly represented around the world. One example I showed is there is a gene that influences the size of the brain a little, not very much. And this gene is not found in Japan because the migration of human populations from Africa did not reach Japan by the time that this mutation was evident. And so I think this becomes probably easy to look at in the context of ethnic versus disease risk, because this is very important to know the incidence. I think, to be very politically correct, people were surprised just how similar the brains of different populations are. So if you look at height, the height of a person, around the world there are very different average heights for different cultures. But many of the properties in terms of proportional brain volume, genes that affect them, were so universally found. There are a few exceptions. So there are some genes that affect the brain and also affect cardiac health risk that are not found in Asian populations, but they are found in Caucasians and also in Africa. And so this is known. I mean, the prevalence of these heart conditions is also different in these societies. But there is some interest in different cultures that perhaps in China the way people do mathematics is different because the language is pictographic. And this could be true. And so maybe we could look at this in a positive way and try and find ways that each culture could be helpful. Thank you. There's maybe time for one more question if anyone has it.
You asked me something, or?
Oh no, we're done. Ничего вопрос. Спасибо большое.
Еще есть вопросы, нет?
Спасибо.
Да.
И Paul Thompson. И now I want to ask you, would you like to tell us something about what is the future of neurotechnology? Okay, we will know all about DNA, for example. We will know about connectomes with high resolution, but then, what we can?
Да, правда, спасибо, профессор Каплан. Правда, я думаю, что это интересный вопрос. Извините, говорю по-русски, по-английски. Очень плохо. Спасибо. I think the question is whether where neurotechnology is going and what are the major applications we can see on the horizon. I wanted to pick up your point in this slide here. One area that we've had some interesting and exciting work is neuropharma. And so if you look on this slide and look at the areas that are being looked at, there's tremendous interest between the pharmaceutical industry and people who collect information from the brain to see what the drugs are doing. And it's changing. And so about 5 years ago, drug companies would come to us and they'd say, can you design a way of imaging the brain to see whether the treatments we're applying are working? And we used to have contracts and consultancies with companies such as Lilly. They were making antipsychotics that were reducing the hallucinations in the brain, neuroleptics that were reducing the incidence of epileptic seizures. They were interested in how the drugs were modulating brain activity. And also in the field of brain aging. And so, I mean, all of us are aging. We're all interested in whether there are vaccines to get rid of amyloid in the brain, whether there are growth factors or things such as erythropoietin that affect blood perfusion to the brain. So 5 years ago, we would often be asked, please can you design a way of imaging people who are taking our new drug versus the old one? And what we used to do first is we'd have to decide how much it would cost to do a well-powered statistical analysis of a drug. And so if the company thought that their drug could reduce the rate of brain aging by, let's say, 20%, we would design a study where you scan them with MRI today. Half of them take the drug, half of them don't, and you'd scan them with MRI in 2 years. And the drug company would say, 2 years is too long. Can you design a method that will look at changes over 3 months? And of course, with structural MRI, the scanner is not particularly stable. You come back in 3 months and your brain looks 1% bigger or 1% smaller. Now, what we have seen in the last 5 years is a huge improvement in imaging technology, and we wouldn't even do that at all today. So one thing that's happened in the field of brain mapping is, many of you will know this, if you go to a brain imaging meeting today, there's a lot of interest in resting state functional MRI. And what you do, it's very easy. You lie down in the MRI scanner, and the fluctuations in blood flow in your brain are observed over a period of 10 minutes. And from this, you statistically reconstruct the connectome, and it's the network of correlations, or the network of synchrony in your brain. And this is a very convenient method of assessing the brain. You don't have to be good at it. There's no task. And from this, you can look at the network of communication. And this has applications in coma, neurotrauma, serious traumatic brain injury, to see if the brain is coming back to life with a treatment or with a surgery, or whether or not a medication has been effective. And I think this is tremendously exciting. It didn't even require a new scanner or a new development in technology. It was just a different way of using the scanners that we have today to look at fluctuations in brain activity. So I think what we're seeing, certainly in the next few years, is an increasing use of ways of non-invasive imaging of the brain to see if drug treatments and interventions, and certainly neurorestoration, as Dr. Zellman mentioned, are effective. And let's hope that they give us some new and promising treatments in the near future. Спасибо большое.
Just one, Paul, how complicated our brain? How complicated our brain? Is it possible to modeling or something?
Artificial intelligence, yeah.
No, like a human brain. Is it possible?
I think that's a great question. I mean, the level of complexity that we're able to comprehend is limited. And so there may be many features that we're unable to see despite them being there. And so I think we're reminded of astronomy. I mean, I think primitive people saw the world as very limited, and now we know that there's much more to it. So perhaps in the near future we'll find a whole new horizon to see beyond what we now see. I think that's a very good question. That may be one of the more tractable questions. And so we have complete information in principle on the sequence of the genome. We lack information on the mechanism, but we have fairly detailed information on brain networks at the level of macrostructure. So it seems like, although we don't yet know, that may be one of the questions that's easier, certainly easier than some of the other questions discussed today.
Пол сказал, что в принципе на самом деле найти связь между генетикой и тем, как мозг организован, может быть проще, чем понять, как мозг работает. Потому как мы можем знать практически все про генетику человека, мы можем получить сейчас полный секвенс генома, и мы с помощью современных технологий можем совершенно неинвазивно получить очень достаточно высокого разрешения карту мозга. И поэтому мы можем просто проколерировать и найти детерминанты, которые за это отвечают.